This is Sales Team 20 coming to you to tell you about this awesome new product that we have in store. It will only cost you $37 and is easy as collecting a blood sample to tell you all about how to increase your herd strength and increase your profitability.
How does a producer genetic test his or her cattle? Genetic testing is relatively easy. There are many ways to collect samples for genetic testing that include sampling hair or drawing blood. In this video, an example of genetic testing by drawing blood is performed. Needed supplies for pulling blood on an individual is a sterile syringe and needle, knowledge of how to draw blood on the animal, and a blood card to deposit blood on. The blood card will then be sent to a company who will analyze the DNA for markers. Marker-assisted selection is a process that enables the accurate selection of specific segments of DNA that are associated with a measurable difference or effect on a complex trait, like weaning weight or marbling score. By using marker-assisted selection, a producer can improve EPDs of a selected individual by understanding the allele frequencies and tendencies for a selected trait that the producer has a desire to enhance. This could mean if the producer selects for enhancing meat quality traits such as marbling, he or she could enhance meat production from a choice ribeye steak to a prime ribeye steak.
Welcome to Zoetis Labs. Here we received the blood card that the producer sent in to genetic test his cattle. But before we discuss what we're going to do with the card and what we'll learn from it, let's learn a few basic principles of genetics. The first thing we want to know is what a genome is. A genome is an entire set of 60 chromosomes since we're speaking about cattle, and that is called a genome. The genome is composed of 3 billion, yes, billion with a B, base pairs. Our base pairs are adenine and cytosine, and guanine and thymine. Here I have a makeshift cattle genome represented by M&Ms. Each color of M&M is based on a base pair. So here we have thymine, guanine, cytosine, and adenine. So what I'd like to talk about when we test the blood card for the DNA is SNPs. This is how we're going to tell what the producer wants to know. SNPs are single nucleotide polymorphisms. A SNP is also a variation in a single base pair. So, when the cell replicates this genome, everything should line up. So when it replicates the cytosine, it will continue to replicate cytosine in this sequence. But when a SNP happens, it's a single base pair substitution. So when something substitutes, our cytosine has now been substituted with a thymine. This changes the sequence and also tracks heritable traits. So, SNPs are the reasons geneticists can help predict inheritance of simple traits like coat color, or in this example, complex traits such as meat tenderness or marbling.
In our population, we will have an array of phenotypes. Variance is a measure of how spread out a distribution is. Take this graph. Most quantitative traits have a bell-shaped curve. On this graph, we are looking at the distribution of phenotypes for ribeye area. Each dot represents 10 individuals. As you can see, there are two extremes. The red dots represent the lower extreme of the population with the smaller ribeye area. On the other end of the spectrum, we can see the yellow dots, which represent the upper extreme of the population that has an exceptionally large ribeye area. The majority of the population is represented by orange dots that have an average-sized ribeye area. Variance is important because it predicts the accuracy of EPDs, BVs, and PTAs.
A great example of why we use the EPDs in the genetic testing is if we have this bowl right here, we can tell the physical characteristics of it. We can tell if it has any abnormalities or if it's sound enough. But what we can't tell is the internal properties of it. So by having an EPD score, we can actually look at this bowl and say whether it is prime, choice, or select just based on the EPD score. So we can go from only being able to see this part to once we have an EPD score, we can actually tell every little part of that bowl.